Okay, for the introduction of genetic variation it is defined as the genetic makeup organism within population change. Genes are inherited DNA segments that carry protein production codes. In alternative forms, chromosomes exist that specify various characteristics that can be inherited from parents to offspring. Genetic variation is necessary in natural selection. In natural selection, organisms with environmentally selected traits are better able to adapt to the environment and pass on their genes. For example, in eye color, blood type, camouflage in animals, and leaf modification in plants. The next is importance of genetic variation. Firstly, it makes some individuals better fitted in the struggle for existence. A population has a better chance of surviving and flourishing than a population with limited genetic variation. Genetic diversity also decreases the occurrences of unfavorable inherited traits. Secondly, species do not remain static. Instead, they are slowly getting modified, forming new species with time. Next is improved races of useful plants for increased resistance, better yield, and others. When a population contains genetics of individuals who vary significantly, some of the individuals in the group can increasing the group's chance for survival when these individuals breed with the others. Last but not least is helps organisms survive in different climates and environments.
Sources of genetic variation. During meiosis, crossing over and random orientation of homologous pairs can promote genetic variation in the gametes produced. This is because crossing over can result in DNA being exchanged between non-sister chromatids in a homologous pair. Random orientation refers to the location of the maternal and paternal chromosomes in a homologous pair when they line up along the equator. In this example here, the paternal chromosomes of each homologous pair are on opposite sides of the equator, but it's just as possible that the paternal chromosomes could have been on the same sides. In reality, the orientation is random, which results in genetic variation in each of the gametes produced. Finally, fusion of gametes from different parents during random fertilization results in genetic variation in the offspring.
Individuals of a species have similar characteristics, but they are rarely identical. The difference between them is called variation. Genetic variation is a result of subtle difference in our DNA. Single nucleotide polymorphism are the most common type of genetic variation amongst people. Each single nucleotide polymorphism represents a difference in a single DNA base A, C, G or T in a person's DNA. On average, they occur once in every 300 bases and are often found in the DNA between genes. Genetic variation results in different forms or alleles of genes. For example, if we look at eye color, people with blue eyes have one allele of the gene for eye color, whereas people with brown eyes will have a different allele of the gene. Eye color, skin tone, and face shape are all determined by our gene. In contrast, although weight is partly influenced by our genetics, it is strongly influenced by our environment. For example, how much we eat and how often we exercise.
PCR, polymerase chain reaction. PCR are technology indispensable to genetic and molecular biology. The technique used to make numerous copies of specific segments of DNA quickly and accurately. It enables investigators to obtain the large quantity of DNA that are required for various experiments and procedures in molecular biology, forensic analysis, involuntary biology, and medical diagnostics. It is carried out in a thermal reactor thermocycler. Application of PCR. First, DNA sequencing. Second, detection of microorganisms. Third, DNA fingerprinting. PCR offers an inexpensive DNA amplification of DNA fragment. It's also known as molecular photocopying. So how does PCR work and what we need? First, DNA fusion, second, buffer, third, primer, and lastly, DNA polymerase.
PCR process. Before the process of PCR start, this is the requirement needed. First, DNA portion, buffer, primer, DNA polymerase, and nucleotides. When the requirement complete, the PCR process will start. PCR sequence divided in 3 steps. First is denaturation, second is annealing, and third, extension. Denaturation. As in DNA replication, the two strands in the DNA double helix need to be separated. The separation happens by raising the temperature of the mixture, causing the hydrogen bond between the complementary DNA strands to break. This process is called denaturation. The second step is annealing. Primer bind to the target DNA sequence and initiate polymerization. This can only occur once the temperature of the solution has been lowered. One primer binds to each strand. The next step is extension. New strands of DNA are made using the original strand as template. A DNA polymerase enzyme joins nucleotides together. This enzyme is often tag polymerase, an enzyme originally isolated from a thermophilic bacteria called Thermus aquaticus. The order in which the free nucleotides are added is determined by the sequence of nucleotides in the original DNA strand. The result of one cycle of PCR is two double-strand sequences of target DNA, each containing one newly made strand and one original strand. DNA cycle repeated many times, as most processes using PCR need large quantity of DNA. It only takes 2 to 3 hours to get a billion or so many copies.
Okay, I will continue with my part. End product of PCR, polymerase chain reactions. Extending state. The final step, the heat is increased to 72 Celsius to enable the new DNA to meet by a special DNA polymerase enzyme which adds DNA base. When the temperature increases to 72 degrees Celsius, which is optimum for DNA polymerase activity to allow the hybrid primers to be extended. The result of one cycle of PCR is two double-strand sequence of target DNA which is contained one newly made strand and one original strand. The cycle is repeated many times, usually 20 to 30 times, as most processes using PCR need large quantities of DNA. It only takes 2 until 3 hours to get a billion or so many copies. The new fragments of DNA that are made during PCR also serve as templates to which the DNA polymerase enzyme can attach and start making DNA. The result is a huge number of copies of the specific DNA segment produced in a relatively short period of time. Okay, the figure shows the illustrations how the polymerase chain reaction PCR produce lots of copies of DNA. Okay, that's all for us. Thank you.